Hello everyone, welcome back to our channel Alpha Doctors, where we are going to continue our section of the microbiology. So in previous section we studied about the gram standing of different microorganisms. Some of them were standing gram positive, those containing the thick peptidoglycan layer, whereas those that do not contain thick peptidoglycan layer, or those which were intracellular organisms or have different specialized characteristics. Those did not stand well with gram standing. Today we are going to study about the properties of the growth media. So we have two types of growth media. One of them is the selective media, and other one is the differential media, or which it can also be said as the indicator media. So the selective media is, as the name suggests, is selective for some particular organisms, which favors growth of that particular organism, whereas inhibiting growth of the rest of all the organisms. For example, we have a Thayer Martin agar that grows the that favors the growth of Neisseria specifically, whereas it inhibits the growth of all the other gram positive and gram negative bacteria. Then we have the indicator media or differential media, which tells a possibility of a range of organisms that grows within a particular characteristics. For example, the E. coli are one of the few bacteria that are lactose fermenters, and all the lactose fermenters and grows pink colonies on the McConkey agar. So E. coli is one of them, but E. coli is not specific to the McConkey agar. Other organisms which produce pink colonies in the McConkey agar include the Serratia, and then we have some fast lactose fermenters such as Klebsiella alongside E. coli. So differential media gives a range of organisms that bear some particular characteristics such as lactose fermentation, whereas selective media is, as the name suggests, is selective for one type of bacteria only. So on the specific culture requirements of certain bacteria, we are going to see a growth culture of the some of the most common organisms, starting with the H. influenza. So H. influenza is an organism that commonly causes flu, and it can cause the pneumonia, it can cause the otitis media or otitis externa or meningitis in different age of populations. So H. influenza is an important pathogenic organism, and it requires chocolate agar to grow. So chocolate agar is a agar that is made up of two different types of factors. That is the factor 5, which contains the Na positive, and then factor 10, which is basically hematin. So H. influenza grows on the chocolate agar, and chocolate agar is made up of factor 5 and factor 10. Alright, so then we have the Neisseria gonorrhea. So Neisseria gonorrhea and Neisseria meningitis is a class of organisms that are classified as the gram negative cocci. So we are going to study about gram positive cocci, gram negative cocci in detail in our future sections, but for the time being, Neisseria gonorrhea and Neisseria meningitis are a group of pathogens that can be characterized as Neisseria, and Neisseria grows on the Thayer Martin agar. So Thayer Martin agar is a very specifically made agar that selectively favors the growth of Neisseria by inhibiting growth of the gram-positive organisms with vancomycin, other gram-negative organisms with the trimethoprim and colistin, and it also inhibits growth of the fungus with this statin. So this is a commonly questioned or commonly tested on the U world, where they value that there is a media which contains the vancomycin, and it contains the trimethoprim and colistin and this statin, and this specialized culture will grow which organism basically. So the answer will be either Neisseria meningitis or Neisseria gonorrhea, depending on the options. So Neisseria is a species that grows on Thayer Martin agar. Then we have the Botryella pertussis. So Botryella pertussis is an organism that grows on specialized charcoal charcoal agar. Basically the agar name is potted gangjo agar. It is basically made up of potato extract. So Botryella pertussis is another important pathogenic organism in young population and young children, which causes the whooping cough, and about its detail we will study in our future section. Then we have the Corynebacterium diphtheria. Corynebacterium diphtheria is a gram-positive organism that grows on the tellurite agar or the loeffler medium, which is characteristic to the Corynebacterium diphtheria. Then we have the Mycobacterium tuberculosis. So about the MTB, we studied in our previous standing video that MTB grows on the uh, LJ medium, that is Lowenstein-Janssen medium, because it contains mycolic acids in its cell wall, for which reason it cannot be it cannot be stand either gram positive or gram negative. So it is usually stand in a special type of growth cultures and growth agars. For example, the LJ medium is the most commonly tested question in the world. Then you have the Mycoplasma pneumonia. Mycoplasma pneumonia is a organism that grows on the eaten agar and it requires cholesterol. So its important feature of this agar is that the addition of cholesterol will favor the growth of the Mycoplasma pneumonia. And then we have certain lactose fermenting organisms that grow pink colonies on the McConkie agar due to fermentation of certain molecules which will produce acid, and acid produce the pink color in the pH range, for which reason these organisms grow pink colonies. So E. coli can also be grown on the EMB agar. So EMB is an eosin methylene blue agar, and in EMB agar the E. coli will grow with green metallic sheen. Then we have for further organisms such as Brucella, Francisella, Legionella, and Pasteurella. So these organisms are basically grown on the agars that contain cysteine and iron. So these organisms are grown in the cultures which basically require cysteine and iron for growth. So and then we have the fungi. So fungi are most often grown on agar type that is named as the saburide agar. So here's a pretty mnemonic that you can remember, that saps are fungi. So saburide agar for fungi. Alright, then here's some anaerobic and aerobic organisms. So the aerobic organisms are those organisms which require oxygen to generate the ATP, as the name suggests, aerobic. These include Nocardia, Pseudomonas aeruginosa, Mycobacterium tuberculosis, and Bordetella pertussis. So it's another reason for the reactivation of tuberculosis in an immunocompromised individual, or those individuals which are on TNF-alpha inhibitors. And these aerobic organisms have predilection for the apices of the lungs where the oxygen concentration is the highest. So aerobic organisms are those organisms that require oxygen for growth. Likewise, anaerobic organisms are those organisms which do not require oxygen for growth, and they can carry out their functional activities such as production of ATP without the need for oxygen. So there are only 4 organisms that are commonly tested on the USMLE that you need to remember. These include the Clostridium. So the Clostridium includes 4 further different species that we will study. So for the time being, Clostridium, Bacterial, Fusobacterium, and Actinomyces. So here you can remember a very good knowledge, that is can't breathe fresh air. So C stands for the Clostridium, B stands for the Bacterial, F stands for the Fusobacterium, A stands for the Actinomyces. Then we have the facultative anaerobes. So facultative anaerobes can use oxygen as terminal electron acceptor to generate ATP, but can also use fermentation. So these organisms are those which can survive in the aerobic as well as the anaerobic conditions. The examples include the streptococci, the staphylococci, and enteric gram-negative bacteria. Then we have some intracellular bacteria, and it includes 2 different types of species, that is the obligate intracellular, facultative intracellular. So the obligate intracellular organisms completely rely within the host cell to favor their growth, including the Rickettsia, Chlamydia, Coxiella, and they rely on the host ATP, that is, they do not generate their own ATP. Whereas facultative intracellular organisms are those which can survive either inside or outside the host cell. These include Salmonella, Neisseria, Brucella, Mycobacterium, Listeria, Francisella, Legionella, and Yersinia pestis. I know these names can be really confusing at this point, But as we move on, you will get familiar with the names, and as we study about the detailed pathogenesis and disease-causing ability of each bacteria, you will be more familiar with their names and the pathogenic mechanisms. Then we have some encapsulated bacteria. Examples include the Pseudomonas, Streptococcus pneumonia, H. flu, type B, Neisseria, E. coli, Salmonella, Klebsiella pneumoniae, and group B Streptococci. So their capsules serve as the antiphagocytic virulence factor. So this is an important point that you need to remember. There's those bacteria which are encapsulated. So the capsule provides a major vigilance factor by inhibiting the phagocytosis uh, through the phagocytic cells such as neutrophils, macrophages, and complement-mediated destruction. Then we have some encapsulated bacteria vaccines. Some vaccines that contain the polysaccharide capsule antigens are conjugated to a carrier protein, which enhances their immunogenicity by promoting the T cell activation and subsequent class switching. So a polysaccharide antigen alone cannot be presented to the T cells. This is an important point to remember, that a polysaccharide antigen alone cannot be presented to the T cells because it will not induce a good immune response. The polysaccharide antigens are always conjugated to a carrier protein to induce a T cell activation response and better immunogenicity against encapsulated bacteria. And vaccines include the pneumococcal vaccine, which includes the PCV13 and PPSV23, depending upon the number of species that it can provide immunity against. Then we have conjugated vaccine against the influenza, such as influenza type B, which is also a conjugate vaccine, and for the meningococcal, we include the meningococcal vaccine. Then we have some urease positive organisms. So urease positive organisms need to remember their names for a basic, common and pathogenic trait which is common to these organisms, which is the urea producing mechanisms. The urea producing mechanism is helpful for these bacteria in producing urinary tract stones, most likely the MAP stones, that is the magnesium, ammonium, and phosphate. So these organisms favor the formation of steroid stones or the magnesium, ammonium, phosphate stones. These organisms include Proteus, Acanthococcus, the H. pylori, Ureaplasma, Nocardia, Klebsiella, Staph epidermidis, and Staph saprophyticus. Here's a very easy mnemonic that you can remember. That is the P chunks. The P stands for Proteus and subsequent organisms with their respective first letters. So these organisms contains urea that hydrolyzes urea to release the ammonia and carbon dioxide, which will increase pH, and increased pH will favor the formation of MAP stones that are also known as the steroid stones. So this is all about today's video. I know it's too much to remember, but there's nothing to worry about. As we will move on, we will learn the specific characteristics to each bacteria and their pathogenesis, and it will get easier time by time. So I hope you understood the basic concept of this video. See you in the next video. Thank you very much.